Okay, it's just about 5 o'clock. So welcome to today's Lipid Maps live webinar. So I'm Bill Griffiths from Swansea, and I'll be the host of today's webinar. So the webinar will be presented by Professor Jeremy Tomlinson from Oxford. So Jeremy graduated from Oxford Medical School in 1995, having previously completed his undergraduate degree at Cambridge. He obtained his PhD from Birmingham, studying steroid metabolism and human obesity. He's now Professor of Metabolic Endocrinology and Consultant Endocrinologist at the Oxford Centre of Diabetes Endocrinology and Metabolism. His research investigates the pathogenesis of human obesity, insulin resistance, and non-alcoholic fatty liver disease. So before Jeremy starts, let me just go through a couple of house rules. So the webinar is being recorded, and then it will be uploaded onto the Lipid Maps website and YouTube channel within the next 48 hours. The webinar will be around 45 minutes, allowing 10 minutes or so for questions at the end. So if you have any questions, please use the question and answer function throughout the presentation. And at the end of the presentation, we'll unmute you to ask your questions. So without any further ado, I pass the floor to Jeremy. Jeremy.
Brilliant. Well, thank you very much, Bill. It's a great pleasure to be invited to give this webinar. And what I'd like to do today is to really share with you a bit of a story, really, as to I suppose ultimately how I ended up giving a Lipid Maps webinar, which certainly wasn't necessarily the plan that we started when we started looking at fatty liver disease many years ago. But I think it tells a story of following the science, and really you can't always dictate where that takes you, and that's always the exciting part of what we do. So. Let's start with fatty liver disease. Now, fatty liver disease, as we know, is a huge epidemic of monstrous proportions. You really only have to look at the lay press really to see that almost on a daily, a weekly basis, there's something that's written about the obesity epidemic and how this might have an impact on our daily lives, and in particular focusing on the liver. And it's really over the last sort of 15, 20 years that we've begun to really understand that very tight relationship between obesity, the impact that it has on the liver, but also the interplay between that and type 2 diabetes. And again, it's trying to unravel the mechanisms that drive that condition and how we can use those mechanisms to not only inform how we can understand and treat the disease, but also to discover whether there are new approaches to the management of patients with fatty liver disease. So many of you will be familiar with non-alcoholic fatty liver disease, but this is the sort of the classic diagram of what we might call the spectrum of fatty liver disease. You start over here on the left-hand side with a healthy liver. But actually, in a significant proportion, perhaps 1 in 3, 1 in 4 people, you end up accumulating fat in the liver. That's non-alcoholic fatty liver or hepatic steatosis, and that's incredibly common, and on its own is relatively benign. It doesn't convey any major problems. But in a certain proportion, perhaps 20 to 30 to even 40%, that fat can drive an inflammatory process, and that's called non-alcoholic steatohepatitis. And that's characterized by various histological features, and those are outlined here. That's steatosis, that's lipid accumulation, as we know, the inflammatory process associated with that. And then this rather sort of peculiar term of ballooning. When hepatocytes become damaged as a result of this inflammatory burden, they end up with a more rounded phenotype, and they're classically called a balloon hepatocyte. And that's really a marker of this non-alcoholic steatohepatitis. That inflammatory burden can then really predispose to developing fibrosis and scarring. And this interplay between steatosis and inflammation is reversible. And we know that you can reverse steatohepatitis through modifications, including lifestyle modification that we'll talk about a little bit later. But importantly, it's this fibrosis, because fibrosis is the key thing that determines outcome. And we stage fibrosis from histologically from 0, which is no fibrosis, all the way through from 1, 2, 3 to 4. Now, 4 is cirrhosis. And you can get cirrhosis from non-alcoholic fatty liver disease in exactly the same way as that you can get cirrhosis from consuming too much alcohol or from a viral hepatitis. And what's important is it's this fibrosis stage, not really the inflammation or the fat, it's the fibrosis that dictates outcome. And generally speaking, the more severe your fibrosis, the worse your outcome. And that's shown data here. This is again is now, I suppose, getting on towards 10 years old. But this is perhaps some of the most robust data. Here we've got histological staging of the liver biopsy, because the standard approach for staging how severe your disease is a liver biopsy. We'll come to that towards the end of the talk. But if you've got moderate or limited fibrosis, so that stage is 0 to 2, then your all-cause mortality is not significantly raised in comparison with a reference population. But once you end up with fibrosis stage 3 and 4, bear in mind that stage 4 is cirrhosis, you have significant increases in mortality, all-cause mortality. And in particular, that's driven by a risk of hepatocellular cancer, HCC, cirrhosis and the complications related to cirrhosis and liver-related mortality. But actually, what is crucial here is that most patients will die of cardiovascular causes, because actually what this is a marker of is a marker of severe and advanced and high cardiovascular risk. So actually a patient with cirrhosis as a result of non-alcoholic fatty liver disease is 4 or 5 times more likely to die of cardiovascular disease than they are of liver-related mortality. And again, that's sort of shown here. If you look at all-cause mortality, this is more recent data. This is fibrosis stage on the x-axis. As fibrosis stage increases here to F3 and F4, you can see that all-cause mortality rises significantly. But also there is an increase in liver-related mortality as well. But the disparity between these two lines is really filled in by cardiovascular outcome. So this is both a liver disease and a cardiovascular disease. But how important, how big a problem is it? People will say in our diabetes clinics, well, I'm not seeing patients with jaundice and very advanced liver disease. But this, I think, is quite striking data published relatively recently. And this tells us about the number of transplant listings. So these are patients being listed for liver transplant. And it's divided up here into those patients that are listed who don't have a liver cancer. So that might be that their liver is failing for whatever reason. And it's all set relative to the numbers of cases in 2002. So it's a 20-year interval here. And what you can see here is that the proportion of transplant listings for liver failure, effectively, has been dramatically increased in our non-alcoholic fatty liver disease cohort in comparison with all the other causes, which are shown in the other colors, of chronic liver disease. And the same really holds true if you have a liver transplant because you've got a primary liver cancer. Again, it's the cause related to fatty liver disease that is increasing so much faster than all the other causes. In terms of absolute numbers, alcohol probably still tops fatty liver disease in terms of being the most common indication for liver transplant, but actually the two lines are merging. So this is a big problem, and you can see here there seems to be no attenuation of the gradient of increase in the rise of NASH as an indication for liver transplant. So there's been lots of excitement, really, about can we develop new treatments. And again, the reason I put this slide up, it's a fairly horrendous slide for a variety of reasons. But the reason I put it up is because it shows the multiplicity of targets that people have been investigating to try and improve patients with fatty liver disease. One of the good things about this diagram, taken from a pretty recent review, is that it's not entirely liver-centric, because what we do understand is that fatty liver disease is actually a multi-system disorder. We know, for example, that adipose tissue has a crucial role, and actually most of the fat that we end up storing in our liver is derived from lipolysis, from fatty acids derived from adipose tissue that enter the portal circulation, then enter the liver. Yes, there are the processes of things like de novo lipogenesis, generating new fatty acids within the a new triglyceride within the liver. There are impairments in oxidation and transport out of the liver. So yes, there are liver-specific processes, but actually you can see that lots of targets are actually being explored in investigating other tissues, not just in the liver. But it's complicated. And actually, in terms of finding a specific pathway that we can intervene with in order to improve fatty liver disease has proved pretty challenging. So how did I first get interested? I'm an endocrinologist and diabetologist by training. So how did I start getting interested in the liver? Well, it all started many years ago, really, with our interest in glucocorticoids. And this is Harvey Williams Cushing, who was a neurosurgeon. And this is the first patient that he described with what we now understand is Cushing's disease. This is Minnie G. And she had a variety of different metabolic complications, including weight gain. You can see the central adiposity here. She had muscle weakness. And if we had an opportunity to look in her liver, I'm sure she would have had fatty liver too. And this is really from 1912. So this is, well over 110 years, 110 years ago. And what we now understand, if you look at the panel on the right, are really the multitude of effects that glucocorticoids have on the body. In fact, I can't really think of an organ system where glucocorticoids aren't really impacting on the system. But in particular, they have pronounced metabolic effects, including predisposition to type 2 diabetes and insulin resistance. We've talked about weight gain, muscle weakness, fatty liver disease, as well as having lots of other impacts, both cognitively, on the immune system, on skin function. So pretty much every organ system in the body. And again, I grew up doing a lot of work trying to understand that link between glucocorticoids and metabolic phenotype. And we initially started looking at glucocorticoid metabolism. In particular, we start off with cortisol, the endogenous glucocorticoid that's produced by the adrenals under the activity of the hypothalamo-pituitary adrenal axis. But we know that actually the metabolism of cortisol locally within tissues by a variety of enzymes, a few of which I've highlighted here, are quite critical in governing the availability of glucocorticoid to bind and activate its cognate glucocorticoid receptor. And we first started looking again, or when we got into fatty liver disease, at the 5-alpha reductases that convert cortisol ultimately to the dihydro and then the tetrahydro metabolites of cortisol, in particular here, 5-alpha dihydrocortisol. So 5-alpha reductase is quite an interesting enzyme. In fact, there are 2 isoforms of 5-alpha reductase. And the isoforms really converted, we've already seen, convert cortisol to the dihydro and then subsequently via 3-alpha HSD into the tetrahydro metabolites of cortisol. And you can see here in the top part of this diagram how that would clear glucocorticoids and prevent them binding to the glucocorticoid receptor. So potentially 5-alpha reductase has an ability to limit glucocorticoid receptor activation. But there's also a flip side to that coin, as many of you will be aware, is that 5-alpha reductase also takes testosterone and converts it to 5-alpha dihydrotestosterone, which is the more potent androgen. So, and that more potent androgen, 5-alpha DHT, will bind to the androgen receptor and regulate androgen gene expression. So you've got 2 things at play here. One is clearing of glucocorticoids, and the second thing is activating of androgens. And we know that there are 2 isoforms of 5-alpha reductase, type 1 and type 2, and they are differentially expressed. So we know that type 2 highly expressed in the prostate urogenital epithelium, but interestingly in the liver, and specifically in human, it's different in rodents, is that both the type 1 and the type 2 isoform are expressed, and in adipose tissue, it's pretty much exclusively the type 1. But importantly, what we also have is a clinician, but we also have clinical tools whereby we can actually manipulate 5-alpha reductase inhibitor. And both these drugs here, finasteride, which is a selective 5-alpha reductase type 2 inhibitor, widely used in clinical practice to treat prostate enlargement and to instigate androgen deprivation in prostate cancer. But there's also, perhaps more recently, dutasteride, which is a non-selective, both 5-alpha reductase type 1 and type 2 inhibitor. And so those gives us tools whereby we can say, well, yes, we can do in vitro, we can do rodent experiments, but can we use those tools in clinical experiments, as I've shown in a moment, to try and tease out what 5-alpha reductases might be doing to the liver? So the first experiments we did were looking at 5-alpha reductase type 1 knockout mice and 5-alpha reductase type 2 knockout mice. Now, the caveat here, which is important to note, is that 5-alpha reductase type 2 is not expressed in the rodent liver, contrasting with humans. But what we noticed in the 5-alpha reductase type 1 knockout mice was that their liver to body weight ratio is increased, their livers were enlarged. And when you look at their livers, you can see here that there was increased steatosis and quantified hepatic triglyceride, you can see in the top panel there. So these animals were predisposed to developing fatty liver. The 5-alpha reductase type 2 knockout mice weren't, but again, not altogether surprising, bearing in mind it's not really expressed in the rodent liver. But we also intriguingly saw that actually when we put these animals on a diet that not only drives hepatic steatosis, but inflammation and fibrosis, that the 5-alpha reductase knockouts were actually protected from the development of hepatocellular cancer. So quite a curious phenotype, but it certainly gave us the first indication that the 5-alpha reductases were important in regulating metabolic and potentially proliferative phenotype in the liver. This is not always. This is work with Dawn Livingston and Ruth Andrew, who also were working on 5-alpha reductase knockout mice, and actually they did a more in-depth analysis on the phenotype. And again, showed similar things in the sense that they showed that these mice were insulin resistant. Here we've got the 5-alpha reductase type 1 knockout mice with elevated insulin levels under a glucose tolerance curve, and that persists not only within 6 months but also 12 months. You see changes on a high-fat diet with insulin action, and also here on the high-fat diet, you can see that liver triglycerides significantly increased in the knockout animals. And using a carbon tetrachloride model, they were able to show again that there was increased fibrosis, this bridging fibrosis that would equate to the most advanced stages that we'd see, so F3 towards F4 on our liver biopsies, and that was more common in the knockout animals. So again, these mice appear to develop an adverse phenotype. But what about humans? Is there any evidence that this could be relevant in humans? And again, one of the approaches that we've always tried to do is to take our observations, be it from cell models, be it from rodent models, and to say, well, is this relevant in a human model? And again, we've often used experimental medicine approaches to do that. And that's exactly what we did here with finasteride and dutasteride. And the hypothesis was that actually, that if you can inhibit the 5-alpha reductases in the same way that if they're knocked out in an animal model, then the hypothesis would be that that would be detrimental to the liver. So we took healthy male volunteers and we did some baseline investigations. We did some magnetic resonance spectroscopy to quantify fat in the liver. We then used clamp procedures, which most of you will be familiar with, but the idea behind those is that you infuse insulin and you try and counteract that insulin with glucose. The more glucose that you have to infuse, it suggests that your body is more sensitive to the insulin because you're trying to keep the blood glucose level perfectly flat. So it's a gold standard way of assessing both insulin sensitivity, but we can also look at glucose metabolism by using stable isotopes to trace the fate of glucose. And then we used adipose microdialysis to sample not only from the circulation, but also from the interstitial fluid within adipose tissue to get a handle on what was happening to lipolysis principally. And then these volunteers were randomized to either treated with dutasteride, that's our non-selective type 1 and type 2 inhibitor, or our selective type 2 inhibitor finasteride. And then after 3 weeks of treatment, the whole procedure was repeated. And to cut a sort of long story short, this is sort of what we showed really. When we looked at hepatic glucose production, again, that's a process that's inhibited by insulin. Insulin normally switches that off. We saw that glucose production in our finasteride, in our dutasteride treated group was elevated. This is post-treatment in the white bars, suggesting that we've got hepatic insulin resistance. And interestingly, we didn't see that with the selective type 2 inhibitor. And when we looked at using our magnetic resonance spectroscopy to quantify liver fat, even after 3 weeks in healthy individuals, we saw that there was a significant increase in liver fat in the dutasteride treated group, but not in the finasteride. Again, the conclusion from that was in a similar way to what we'd seen in rodents, and again, there's cell model data that I haven't shown, that actually this combined inhibition of both 5-alpha reductase type 1 and type 2 is detrimental to the liver. So that was 5-alpha reductase, but actually what we then got more interested in, and again, this sort of brings us back towards how I eventually ended up doing a lipid maps webinar, was actually our interest in 5-beta reductase. Now, 5-beta reductase is this enzyme, which is also crucially important for inactivating glucocorticoids, generating these 5-beta dihydrometabolites of cortisol and also cortisone, which again is an inactive glucocorticoid metabolite. But importantly, it also has other actions. So it is pretty much responsible for the inactivation of the majority of glucocorticoids. And it does that by introducing this sort of almost this 90-degree bend within the structure of the glucocorticoid to prevent it binding and accessing the glucocorticoid receptor. Now, we also know, however, that it has a crucial role in other enzyme systems, and in particular in bile acid synthesis. And again, this is something that I'm going to really talk a little bit more about in the second half of the talk. So we've already told you a little bit about the steroid hormone metabolism, taking cortisol and inactivating cortisol as well as cortisone. But here it's important that it takes substrates. And again, these are substrates that I'm going to talk about a little bit later. These are the 7-alpha, 12-alpha cholesterol and the 7-alpha cholesterol are important, clearly, substrates in generating the primary bile acids, quinodeoxycholic acid and cholic acid. So AKR1D1 or 5-beta reductase is a really crucial enzyme in these two parallel systems. And that got us sort of quite interested in that it might be important for regulating metabolic phenotype in a fairly profound way. So what do? Well, we stuck with what we knew best to start with, and we thought, well, let's just confirm what this enzyme does in glucocorticoids. I think glucocorticoids was always our safety net, our comfort blanket. And interestingly, if you knock down AKR1D1, here we've got an in vitro model where it's a HepG2 cell, actually a liver cell line, where we've knocked down AKR1D1. We've successfully done that. You can see it on the Western blot here, and you can see here in the knockdown, cortisone clearance is not as dramatic as it is in the scrambled control. So cortisone persists, and that's exactly what you would expect. You haven't got as much AKR1D1, you're not clearing cortisone, and in the same way, you're not generating this tetrahydrometabolite because you've knocked down the enzyme that's partially responsible for its generation in comparison with the scrambled control. So it's doing exactly what you might expect in a knockdown experiment. What about an overexpression, the sort of parallel experiment of that? Well, again, here we've used cortisol as a different glucocorticoid. But what you can see when you overexpression here in gray, is that you can see a dramatic increase in the clearance of cortisol within 24 hours, such that you've pretty much got rid of all of the cortisol that you had to start with, and a parallel increase in the generation of your 5-beta tetrahydrometabolite of cortisol. But this enzyme system is also important, perhaps to a lesser degree, but for clearing synthetic glucocorticoids. Prednisolone is by far the most commonly prescribed synthetic glucocorticoid. And you can see here that actually after 24 hours in our overexpression, we've enhanced the clearance of prednisolone in comparison with our empty vector, not quite to the same extent as we saw with cortisol, where we completely got rid of almost all the cortisol, again, similar concentrations to start with. We cleared almost the total of that 400 nanomolar cortisol at 24 hours, but we've only got rid of about 30 to 40% of that, the prednisolone. But importantly, this does have a knock-on effect. So this is a glucocorticoid receptor activation assay, a luciferase assay, and we can see here that by overexpressing and clearing cortisol, we limit that GR activation. We do it to a lesser degree, as you might expect with prednisolone, mirroring what we saw with prednisolone clearance. But interestingly, with dexamethasone, this enzyme system doesn't appear to be important for clearing dexamethasone. So good evidence that actually within our comfort blanket of glucocorticoids, AKR1D1, 5-beta reductase, is having an important role. But really for the latter part of this talk, I want to focus on its role in bile acid synthesis, because AKR1D1, as we've said, is a crucial enzyme in regulating bile acid synthesis. And there are very rare patients that have monogenic AKR1D1 deficiency with mutations in AKR1D1 that develop a life-threatening and potentially very serious neonatal cholestatic jaundice that can result in liver failure. And as you can see here, AKR1 here sits within this pathway, and the substrates for AKR1 that we're going to talk about a little bit more are the 7-alpha hydroxycholesterol and the dihydroxy 7-alpha 12-alpha dihydroxycholesterol. And also this oxysterol here is 7-alpha hydroxycholesterolic acid, or 7-HOCA. And those are important substrates that I'm going to come on to a little bit later. But as you can see, this is all crucially important for the generation of your primary bile acids, cholic acid and quinodeoxycholic acid. So let's go back to our interest in non-alcoholic fatty liver disease. And again, this is the diagram that you've already seen. But is there any evidence that actually these enzymes that I've talked about are dysregulated in fatty liver disease, in particular AKR1D1? Could that possibly be important in fatty liver disease? Well, clearly is, else I wouldn't be sitting here talking to you. So if you look in liver biopsies and you look at gene expression levels, with increasing steatosis, what we see is decreasing AKR1D1 expression. The NAS score is a histological score based upon steatosis, inflammation, and ballooning. So it gives a marker of NASH, increasing scores, more NASH effectively. And you can see here that as you go with an increasing NAS score, your mRNA expression in liver biopsies decreases. What about fibrosis stage? Again, we said that we score fibrosis from 0 to 4. In those patients with most advanced fibrosis, 3 and 4, AKR1D1 expression is decreased. And finally, if you data mine and look at in publicly available databases in patients with hepatocellular cancer, AKR1D1 expression in hepatocellular cancer is markedly downregulated in comparison with healthy liver. So certainly across the board within the context of non-alcoholic fatty liver disease, there's very good evidence now of decreasing expression and activity of AKR1D1 with advancing severity of disease, be that fat, inflammation, fibrosis, or even the development of cancer. So on the back of that, Nick Nicolau, who was originally a PhD student, but subsequently a postdoc who worked with me for many, many years, began to explore the role of AKR1D1 in cellular models and undertook some gene silencing and RNA sequencing. And you can see here the volcano plot of his RNA sequencing knockdown experiments. And when we did the pathway analysis, you can see here we've successfully knocked down, you can see we've successfully knocked down AKR1D1, which is always quite reassuring as the most downregulated gene. But when he did pathway analysis, there's a few things that just lit up, which I think were interesting. And in particular, when you do the pathway analysis, metabolic pathways, insulin signaling, fatty acid metabolism, all really leapt out as being of interest to us, bearing in mind we were exploring this in the context of fatty liver disease. So then what Nick did, again, some of this is published, some of this is unpublished data, but what Nick then did was really explore the cellular processes that might govern its role within the context of fatty liver disease. So he looked at de novo lipogenesis. That's the synthesis, as you'll be aware, of fatty acids from non-lipid substrates. And again, to the critical enzymes that are important in regulating that process, fatty acid synthase, lactyl-CoA carboxylase 1, markedly upregulated at the mRNA level. Also, the protein, haven't shown you that data, but also in the AKR1D1 knockdown cells. Using deuterated water, again, this was done in collaboration with Leanne Hodson in Oxford. Using deuterated water, you can then get a functional readout of de novo lipogenesis, and you can measure the incorporation of that deuterated water into newly synthesized palmitate, and again, significantly elevated in the knockdown cells. And when you look at the intracellular triglyceride, again, higher in the AKR1D1 knockdown cells. There was also an inflammatory burden, so more fat, but also more inflammatory burden. Again, here you can see the gene expression really for many of the pro-inflammatory cytokines. Again, when we measured some of these in ELISAs in the cell media, they were also elevated. So we've got fat, we've got inflammation, but we also have apoptosis as well. Again, 3 classic apoptotic markers here, as well as in the green graph here, marker of apoptotic activity with a caspase 9. So we've got really driving apoptosis within these cells, suggesting that these cells are pretty unhappy with fat, inflammation, apoptosis. And finally, what Nick did was to look at DNA damage. Now, this is something called a COMET assay that many of you may be familiar with, but what it allows you to do is it looks for damaged DNA, and damaged DNA, when you electrophoresis this, comes as effectively as a long tail within the COMET. And here you've got a positive control here with hydrogen peroxide. You can see it's got a big, long green tail here. And what hopefully you can convince you of, I'm not sure how well it projects really, but you can see that this, with the gene silencing with AKR1D1, we have this prolonged tail suggesting there's marked DNA damage in comparison with our scrambled control. And again, you quantify that tail here, showing that we've got a significant upregulation, meaning that we've got more DNA damage in our knockdown cells. So overall, more fat, more inflammation, apoptosis, DNA damage, all of which is pretty bad news, I think, if you're an hepatocyte. But we also began to probe some of that data a little bit more, and this is all of this is unpublished, because actually, when you look at some of the gene ontology pathways, it was quite clear that actually that cell cycle, cellular senescence were all quite important. And again, bearing in mind that we know that the data about fatty liver disease driving cancer risk, that began to get us thinking about, well, could this be involved in cellular proliferation? And again, many of you will be familiar with cell cycle, but this is the sort of cell cycle. It's a very tightly and complicated process, such that you have a whole series of these site-independent kinases that are really important for driving this whole process of cell cycle and cell division. And that these site-independent kinases, these CDKs, ultimately result in phosphorylation of this protein called RB. But the whole process is also tightly regulated by inhibitory proteins, in particular P21, P15, P16. So you have combinations of inhibitory proteins as well as proteins that drive cell cycle. And what was interesting when Nick looked at this was actually that what you see, yes, we've successfully knocked down AKR1D1. Again, that's always very satisfying. But actually across the board, P21, P15, P16, these are these inhibitory proteins were all markedly upregulated. And the site-independent kinases, CD1, CDK2, CDK4, CDK6, that are driving cell cycle were downregulated, suggesting that we might be seeing inhibition of cell cycle or an arrest of cell cycle progression. And actually, when you do flow cytometry, that's exactly what we did see. This is our scrambled control. And here we can see the proportion of cells in G1, S, and G2 phase. When we look at our AKR1D1 knockdown, we see that a lot of the cells are really stalling within S phase. And there's this impaired S to G2 transition, suggesting that we have got this cell cycle arrest. So all in all, we've got evidence that we've got fat, inflammation, we've got apoptosis, DNA damage, and we've got this cell cycle arrest within our AKR1D1 knockdown cells. But the question is, well, how is all this working? How can this possibly be acting? And so Nick and I, we came up with this model that was suggesting that, broadly speaking, you have cholesterol going for oxysterols to generate your primary bile acids. And in a very simplistic way, if you take out AKR1D1, what you might expect to see is increasing of your oxysterols, which you know are potent LXR ligands, and you would expect to see perhaps a downregulation of FXR because you're depleting your cells of their primary bile acids. So, is there any evidence to suggest that may be true? Well, the answer is yes, there is. So when Nick looked at using again a luciferase LXR activation assay in the knockdown cells, we see significant increase in LXR activation on the basis of this luciferase assay, suggesting that we have got LXR activation. Similarly, with when we look at key target genes, here I've already shown you about the regulation of fatty acid synthase, and again, you can see here, this is it's a slightly complicated graph because here we've used various agonists and antagonists of LXR and FXR. So just in the knockdown, I've already shown you that you get increased expression of fatty acid synthase, and that's reflected in the black bar here. The bar next to it uses this KW4064, which as many of you will know is an FXR agonist, and you can see here that completely blocks that induction of fatty acid synthase, suggesting that we can ameliorate that effect by FXR agonism, suggesting that that's really quite important in driving that process. But what's also interesting is if you use LXR antagonists, you can also ameliorate that rise in fatty acid synthase, suggesting that perhaps there's a combination here of this LXR agonism combined with antagonism or limited FXR activation that's driving the changes in gene expression. And we see a very similar process for acetyl-CoA carboxylase 1. So again, combinations here of LXR activation and inhibition or limited FXR activation. So can we probe that a little bit more? Well, again, this is a pathway that you're all very familiar with. And again, the three substrates that I'm going to talk about, the 7-alpha hydroxycholesterol, 7-alpha 12-alpha dihydroxycholesterol, and the 7-HOCA, the 7-hydroxycholesterolic acid, all of which are the direct substrates upstream of AKR1D1. And when Nick looked at this is work that he did in combination with Carolyn Cummings over in Toronto, when Nick looked at intracellular oxysterols in our AKR1D1 knockdown, pretty much as you might expect, we did see elevation of both our 7-alpha, 7-alpha 12-alpha, and the 7-HOCA, exactly as you might expect from these yellow substrates over here. And again, as we've seen, if you actually, we know that AKR1D1 knockdown resulted in LXR activation, but when you just apply these substrates, 7-alpha and 7-alpha 12-alpha dihydroxycholesterol were both direct activators of LXR, and you can see that here on our LXR luciferase assay. Nick then did a further assay with both an LXR alpha and LXR beta, and they appeared to be selective LXR alpha agonists, in contrast with perhaps some of the more traditional, for want of a better word, oxysterols, the 27-hydroxycholesterol, which was activating both. So selective LXR alpha agonists. Interestingly, 7-HOCA was not, and we'll come back to 7-HOCA in a minute. But certainly 7-alpha hydroxycholesterol and 7-alpha 12-alpha dihydroxy were potent LXR alpha agonists. So what's the relevance of this? Can it be of relevance to patients with fatty liver disease? Could these novel oxysterols be important for generating the fatty liver phenotype? So this is the graph. The top graph here is the one you've already seen. This is the pattern of oxysterols that we saw with our AKR1D1 knockdown. And again, this is data from Bill and the team, where they have actually looked at oxysterols within liver biopsies from patients that have got NASH. And again, what's interesting here is that it mirrors what we see very closely in our AKR1D1 knockdown. So, for example, here, 25 hydroxycholesterol, where we saw there's no difference in the liver biopsies, but actually 7-alpha, 7-alpha, 12-alpha markedly elevated in patients with NASH. So the pattern that you see with AKR1D1 knockdown, bear in mind that we know, and this is data you've already seen, AKR1D1 goes down with increasing severity. You can imagine that there's a hypothesis that you can generate that shows that downregulation of AKR1D1 translates to elevation level of these oxysterols here, which appear to be elevated in patients with NASH. And that is quite an exciting tie-in from cellular models all the way through to liver biopsies from patients with moderately advanced liver disease. Again, this is unpublished data from Nick and from Bill and Yukin and Swansea. And again, we can then begin to say, well, can we sort of translate this to the clinical setting more broadly? So we've got a cohort of patients in Oxford of who have very advanced liver disease cirrhosis, but also have cirrhosis and hepatocellular cancer. So hepatocellular cancer in the vast majority of cases arises on the background of cirrhosis. And so it's important that you match those controls correctly. Comparing HCC against a person with normal liver is not necessarily appropriate control. But what you can see here is that in patients with cirrhosis and in patients with HCC and cirrhosis, 7-HOCA levels are significantly elevated. In fact, they're about twice what you might expect. When you take healthy individuals, and again, this is something from something called the Oxford Biobank, where we have about 7,000 or 8,000 patients that have had fairly deep phenotyping, including DEXA scans and metabolite profiles. And again, on a metabolite profile measuring 7-HOCA levels, there's a strong positive correlation between your visceral android fat mass, which we know is a known metabolic risk factor, in particular for driving fatty liver disease. As your fat mass increases, your 7-alpha HOCA levels increase. And again, the final graph over here from Bill and Yukin is both your 7-alpha HOCA levels and your 7-alpha hydroxycholesterol levels. And you can see, again, a very striking positive correlation of both those oxysterols with increasing body mass index, suggesting that there appears to be a relationship between these oxysterols and adverse metabolic features, be it cirrhosis, be it visceral fat mass, or be it body mass index, all of which we know are associated with adverse metabolic outcome. So then just thinking a little bit more, what do we know about 7-alpha HOCA? What can it do to cells? So again, what Nick's done here, again, this is unpublished data, is actually he's done some RNA sequencing whereby he's treated HepG2 cells with 7-alpha HOCA. And again, you'll be familiar, this pathway analysis looks very similar, as you might remember, to the AKR1D1 knockdown, in the sense that cell cycle, cellular senescence were all very high up that pathway. And again, you'll remember the diagram that I've already shown you with these cyclin-dependent kinases, the phospho-RB and the P21 inhibiting this cell cycle progression. And that was all what we saw with AKR1D1. But interestingly, what we see with 7-HOCA actually replicates that very, very, very nicely, suggesting that this could be very important in manifesting the effects of AKR1D1 knockdown, because we see with HOCA treatment, this is HOCA treatment, not AKR1D1 knockdown, upregulation of P21, which is what we saw with the AKR1D1 knockdown. We saw downregulation of all the cyclin-dependent kinases with AKR1D1 knockdown. We see it also with 7-HOCA treatment. Important, again, I don't think I showed you it previously, but again, on Western blotting here we've got the phosphorylation of this protein here, phospho-RB, downregulated by HOCA treatment. Again, we've seen that with AKR1D1. And again, you'll remember that comet assay that we saw with AKR1D1 knockdown with DNA damage. And again, here, similar sorts of pattern. Here we've got our positive control with our hydrogen peroxide. And again, this extended tail of damaged DNA that you see with the 7-HOCA treatment, again, quantified here, replicating many of the features that we had with AKR1D1 knockdown. So let's again think about, can we begin to translate this any further? And again, that's cellular models. Are they really relevant to humans? Well, the first step in that was to generate a knockout model. And up until we published this data, there'd been no animal model of AKR1D1 knockout. And again, this is work done by Laura Gathergold, who worked for me for many years. She's now a senior lecturer at Oxford Brookes, and we continue to work collaboratively together. And the phenotype of the AKR1D1 knockout is complex. But what they do have, this is a very sort of pretty diagram of the bile acid composition. Bile acid levels are dramatically downregulated with an AKR1D1 knockout, as you would expect, because it's critical in bile acid synthesis. But the composition of the bile acid is also changed dramatically. And again, that's shown in the colors here. And you can see that sort of quite nicely how there are different colors and different proportions of those different bile acids that we've measured are different in both the male and the female knockout mice. And the 7-HOCA levels are elevated, again, consistent with a knockout and elevated substrate levels beyond that. This is a diagram on the right side here of a form of sort of principal component analysis. And I think what you can see very clearly are the differences in groups between the wild type and the knockout, both in the male and females. And you get these clustering of the white, the black, the red, and the blue in quite discrete clusters. So again, that I suppose is a graphical representation really of the different composition of the bile acids that we've got. But again, I just want you to remember that, because that's something we're going to come to when we think about, can we translate some of this to human studies? The phenotype of this mouse is complex, and there are elements of this that I confess that we still don't understand. But actually, when you look at the circulating lipid profiles, we saw dramatic elevations of monoacylglycerols and diacylglycerols. You can see that here with our metabolo analysis, as well as elevated non-esterified fatty acids. So what we might perceive as quite an adverse metabolic phenotype, with lots of circulating fatty acids and lipid precursors. And we certainly saw circulating triglyceride levels were elevated, and intramuscular triglyceride levels were elevated. But somewhat to our surprise, and we still don't quite have the explanation for this, I confess, was that hepatic triglyceride was actually slightly lower in our AKR1D1 knockout in comparison with our wild type controls, accepting that it was quite a wide spread of total triglyceride content. But certainly within the circulation, what you might perceive as quite an adverse lipid profile. With this model, though, what we also did was try to stress this animal by putting it on a diet that really predisposes and tries to drive fatty liver disease. And that's something called the ALIOS diet, the American Lifestyle-Induced Obesity Syndrome diet, that has trans fats, it's sedentary, it has fructose. And if you give it to an animal, it will develop fatty liver disease. It will develop fibrosis and scarring and inflammation, and will develop liver cancers, in contrast to sort of the more traditional high-fat diets. And when you put the AKR1D1 knockout mice on that diet, hepatic triglyceride increases. Interestingly, we didn't see a change in inflammation, but we did see more fibrosis. And you can sort of see that here on the histology with this Picoceres red staining here, showing the increased fibrosis, and here just on the H&E staining, the white lipid globules, suggesting that, again, these animals here, when you stress them with an ALIOS diet, do get more advanced liver disease. So in the final couple of slides, it's, well, can we take any of these observations and make a difference to the patients that I sort of see week in, week out in the fatty liver disease clinic that I run with the liver specialists? So again, you'll be bored of this diagram now. You've seen it several times. But again, this is the spectrum of liver disease. And I said right at the beginning here that the gold standard way to determine whether you are at this end or at this end is to do a liver biopsy. And that's because your liver test, the traditional liver tests that you might go to your doctor and have, are fundamentally useless. If you take a patient with cirrhosis and you can flip a coin, 50% of the time their blood test will be entirely normal, 50% of the time they'll be abnormal. So doing a simple blood test doesn't help us. There are other ways of doing combinations of blood tests, sometimes with imaging, but again, they've got variable performance, so they're not perfect. And I say the gold standard is the liver biopsy. Now, liver biopsy is not something you necessarily want to have done. It requires a day off work. You have someone sticking a needle into your liver. It has a morbidity, and I confess it does have a mortality associated with it. you selectively sample about 1/50,000th of the liver. And if you actually ask patients, do they like having a liver biopsy? Not surprisingly, patients say, oh, well, I'd rather not if it could be avoided. So there's a big move to say, well, can we develop tests that avoid doing a liver biopsy? So you'll have seen this slide again. This is sort of going back to our comfort blanket of glucocorticoids. And here I showed you a few selected glucocorticoid metabolites, but as you all know, as well as I do, it's a lot more complicated than that, because actually glucocorticoid metabolites, again, we've spent a lot of time looking at glucocorticoid metabolites in the urine. You can see all those shown in yellow here, and that's only a part of the constellation. And again, you can measure these, as you know, with mass spectrometry approaches. And again, endocrinologists love measuring urinary steroids. But what is often lost on endocrinologists, and perhaps was lost on me until many years ago, was that these steroid metabolites are by and large generated by the action of enzymes within the liver. And in particular, the A-ring reductases, in particular 5-beta reductase, is crucial for generating many of these metabolites. So we went in with a hypothesis that actually the constellation of urinary steroid metabolites that you have could actually be not a reflection of what's coming out of your adrenal gland, but really what's coming out of your liver and the state of your liver. So this is the sort of traditional diagram that we all see. And, you know, as an endocrinologist, we love having glucocorticoids, mineralocorticoids, androgens. That's what gets us excited. But we often forget that actually it's the liver that is quite crucial in determining all of these different steroids, and whether they are produced both in quantitatively and qualitatively different could well be dependent upon the state of your liver. And that may give us a good reflection of what's happening in terms of the stage of your fatty liver disease. And to help us with that, we got the help of some computational scientists and machine learning. And these were colleagues in Groningen, Michael Beale in Groningen. And again, we used a form of machine learning that he'd developed called generalized matrix learning vector quantization. And this is my non-machine learning expert take on this, because we wanted to ask two questions, really. Well, it was a two-problem question. You know, have you got advanced disease or have you not got advanced disease? And the way I've often looked at this is actually, you know, it's a bit like saying, you know, can we, based on common features, distinguish between male and female? For example, men tend to be taller than the females. Men tend to have shorter hair. Men tend to have more facial hair. So there's a prototype male and a prototype female. And then when you're faced with an unknown, you can say, well, does it more closely resemble the prototype male or prototype female? And that then allows you to say, well, actually, my unknown is more like an advanced fibrosis or more like a mild fibrosis. And that was the sort of approach that we used. And again, by looking at all the 32 different steroids, we were able to, with reasonable efficiency, distinguish between moderate, mild fibrosis and advanced fibrosis. And here you can see the ROC curves here. Again, you'll all be familiar with ROC curves. You know, area under the curve of 0.5 is like tossing a coin. And here we're up with our GMOVQ approach of 0.92. The FIB4, that if for people that aren't familiar with FIB4, is one of the more commonly used non-invasive tools. It actually doesn't usually perform that well, and we were a little bit surprised how well it did perform. But even so, we were still a little bit better than the FIB4. And when we wanted to say, well, if we take this as a screening approach, can we identify advanced disease in comparison with healthy controls? Then again, the test was really very, very good with an area under the curve of 0.99. And again, we're now in the process of validating this in a prospective cohort. But finally, you'll remember this figure that I showed you, with about the clustering of bile acids in our AKI1D knockout mice, and how they clustered into these discrete groups dependent upon their genotype and their sex. So the question is, well, can we use not steroids, but can we use bile acids and oxysterols to try and look at the stage of fatty liver disease? And again, this is very preliminary, unpublished data, again in combination with Michael Beale, because we've now said, let's put steroids to one side. What about bile acids and oxysterols? And here we've just got a limited profile just of 17. But what you can see is that there's very clear separation between our controls in light green, those patients with cirrhosis in red, and those patients with hepatocellular cancer on a background of cirrhosis in blue, such that with excellent performance at being able to distinguish our controls versus cirrhosis. And actually, you know, bearing in mind this is a small cohort, it's very much the discovery cohort, you know, a promising area under the curve, being able to distinguish HCC from patients with cirrhosis. So let me just begin to wrap up in the next minute or so. We've sort of begun to generate this model in the sense that we now think that AKR1D1 really does have quite a crucial role to play in the pathogenesis of non-alcoholic fatty liver disease. And that decreased expression alters our oxysterol profile that then ultimately results in activation of LXR, but also limiting FXR. And that has a multiplicity of gene transcription effects, altering DNA damage, proliferation, apoptosis, and lipid accumulation. And what I've hopefully shown you is that actually that the precise nature of the oxysterol profile is quite important for determining that cellular response. And finally, there's the potential that actually that those oxysterol profiles could be quite useful clinically, because we might be able to take a patient, not have to do a liver biopsy, simply do a blood or even a urine oxysterol profile, and that will say, yes, you've got mild disease, or no, I'm afraid you've got advanced disease, and that will then govern the management approach that you might take with that patient. Finally, always the most important slide. Lots of people have done all this work, and again, we've managed to assemble a great team of individuals nationally, internationally, and it's been a pleasure to work with them across specialities. And that's one of the beauties is that actually, working with hepatologists, with computational scientists, with steroid biologists, and the two people I really wanted to highlight who have worked with me for a very long time now, something moved on, is Nick and Laura, because clearly without them, as I think I've shown you there, none of this work would have been possible. So thank you all very much for listening. I'd be delighted to take any questions if you have any.
So thank you very much. Thank you very much, Jeremy. That was a lovely talk, educating us all about fatty liver disease, and of course I like to see you bringing out the importance of oxysterols in the disease. So we're open for questions. Perhaps the first question is I think is from Hong Yin. I think you asked a question on the Q&A. Are you there, Hong Yin? Okay, there you are. I can see if I can unmute. No, I can't unmute you. Okay. No, I can't unmute you. So let me ask your question to Jeremy. So I think we're talking about the 5-alpha reductase part now. So did you see fatty acids increase in the 5-alpha reductase knockout model?
So the answer, simply measuring total fatty acid levels, then we didn't see a change. But what we didn't do, partly because it was worked on some time ago and we didn't quite have the resources, was to do a very detailed sort of metabolomic or lipidomic profile. So it would have been nice to do. My reservation, I have to say, about many of the rodent models, and again in particular with these A-ring reductases, is they're not perfect models of human biology. And again, I suppose that was highlighted with the fact that you have type 1 and type 2 in humans, but only type 1 in rodents. And again, there are subtle differences with the AKR1D1 as well. So we didn't do that. And again, we didn't see any we didn't see an inflammatory phenotype either with the 5-alpha reductase model.
Okay, thank you, Jeremy. See if I can unmute the next. No, I can't unmute. Sorry, I can't unmute the next questioner. So I'll move on to the third question. Who's written it out. So, have you studied the link between liver cells, polyploidy, and lipid metabolism in NASH and the development of cirrhosis? That's the first part of the question.
So the answer to that is no, we haven't actually. I mean, it's a good point, and there are lots of things that we don't understand. I mean, I think there are people that have looked at that in the context of NASH, but certainly we haven't, and we haven't done that in the context of that link between the inflammatory drive to NASH or even the proliferative phenotype. But it's a good point, and perhaps something that we ought to be doing, but we haven't.
Well, the second part of the question is, can an increase in hepatocyte polyploidy during the development of liver disease continue to change in lipid metabolism and inflammation?
So it's a really good question, actually, and it speaks to a variety of things, really. So one of the key challenges that we face in fatty liver disease is that actually some people progress and some people don't. Some people develop liver cancer and some people don't. And it is possible that I suppose that actually that polyploidy could actually be important in that. But at the moment, we don't really have very good models. You know, in order to assess that clinically, you have to think about things. Well, do we end up doing multiple liver biopsies? And people don't want to have one liver biopsy, let alone having multiple biopsies. And then you have the limitations of cell models, and a lot of the cell models that we use to study that, and again, part of our work as well, they're cancer cell models, so they're not ideal. So part of the problem we face in trying to work out how changes such as polyploidy might be important in disease progression and development are that the models that we have are difficult and challenging.
Okay, next question. In the knockout mouse liver, there was no TG enhancement. Did you measure cholesterol esters?
Good question. I don't think we did measure cholesterol esters, actually, if I'm honest, but maybe we ought to go back and look at that. I don't think we did, if I remember rightly. I'll have to go back and check, but yeah, so it's a good point, and we ought to. Okay.
Unfortunately, I'm not able to. I'm not unable to mute anybody, so I have to continue asking these.
I think. Can I talk?
Oh yeah, you can talk.
It's Clarissa Maia Monteiro here. So I made this question about the cholesterol ester that I think it will be enhanced because you have already more lipid accumulation. But my other question is about, is there any indication of polymorphisms in the gene of AKR1D1 that could predispose to liver disease?
So another really good question again, and we've looked hard, and it's interesting when you look at fatty liver disease, because there are some genetic polymorphisms that do predispose. They're PLA3, M7, so, you know, so there are a number, but it's relatively small, I suppose, if you compare it with, you know, things like, you know, weight, diabetes, those sorts of things. You know, it doesn't worry me that we haven't seen it, and again, we have looked at it and we can't find it, partly because, you know, some of what we're looking at is not necessarily expression, or, you know, there are other mechanisms that might be at play here in terms of what regulates AKR1D1. Again, data I didn't have to show you here suggests that fat itself might be able to downregulate AKR1D1. And so it may be that, for example, you accumulate fat and there may be something that drives that. You know, again, that could be a sort of PLA3 sort of mechanism. That then allows you to accumulate fat. That downregulates AKR1D1, drives your oxysterol accumulation, fueling the lipid, the liver damage. So we don't find a genetic signal for AKR1D1, but it doesn't dampen my enthusiasm.
Yeah, but then could it be that there is some epigenetic control, because we know that some conditions in the childhood and even during the intrauterine can predispose to liver disease, and so could it be that there's epigenetic modification of AKR1D1?
Absolutely could be. Again, hasn't ever been looked at, I have to say. So, but you're absolutely right. It could be. Yeah.
Oh, thank you. Han Ying, can you speak?
Yeah, can you hear me?
Yeah, very good.
Oh, good. Okay, I was just curious. Did you measure the ceramides in the AKR1D1 knockout mice?
So
in circulation.
Yeah, so the metabolite analysis that we did was part of a broad metabolome analysis, and they did measure a few of the ceramides, but they weren't differentially regulated. But again, it wasn't a comprehensive, you know, untargeted approach. So it's quite possible that there may have been some species that we didn't pick up. So, but on the ones that we did look at, where they weren't different.
Yeah, are those launching ceramides that did you ratio them? For example, ratio the 18-1, 18-0, 16-0 to the 24-0, I think, or 24-1. Yeah, and that's the ratio that Mayo Clinic Mayo Clinic use, because the many in multiple epidemiological studies, these ratios indicates the increased elevated mortality in the cardiovascular disease, and the research has shown that NASH patients are also associated with elevated cardiovascular risk.
Yeah, no, I don't. We didn't look at those ratios in the rodent model, but I think it's a good point, and perhaps we should go back and actually look at that, because again, whilst we were slightly disappointed by the lack of liver specific phenotype, the circulating markers just at absolute levels were quite striking. But I will go back and look at those the ratios. I think it's a good point.
All right, thank you.
Yeah, Christina, would you like to ask a question?
Yeah. Hi, I was told yesterday.
Christina?
Yep.
You can hear me?
Yes,
Okay.
yeah, I can hear you.
Let me ask Christina's question for you. First of all, she says, great talk. Then, do you think FDA approval of obeticholic acid is a proof of concept for your hypothesis?
Yeah, another great question. So for those of you, so obeticholic acid is an FXR, modified FXR agonist. It's probably, at the moment, we don't have any drugs that are licensed to treat fatty liver disease. Obeticholic acid is probably the closest one that we have to getting a license. There's evidence, again, from now phase 3 studies that it can limit fibrosis. And you're right, I suppose, here, if we're saying that if you've got downregulated AKR1D1, limited FXR activation, by restoring that, by giving an FXR agonist, is that helping? So yeah, I mean, I would love to sort of swing it in that direction, saying absolutely that is a mechanism that would actually be entirely consistent with AKR1D1 or limiting AKR1D1 activity as a sort of pathological mechanism that could be driving things. So yeah, absolutely. Clearly, there are other things that could well be at play here, but you're absolutely right. By agonizing FXR would ameliorate AKR1D1 knockdown to a certain degree, exactly as we did in our cell models.
Okay, we seem to be running out of questions, and we seem to be at 6 o'clock. So thank you very much, Jeremy. That was it was a lovely talk, and thank you everybody for coming to the seminar. The last thing I should remind everybody about is the next Lipid Maps seminar is February the 28th, and it's Dieter Stein-Hilber, and that's at 5 p.m. So 5 p.m. on February the 28th. That's when we'll see each other next. Okay. Finally, thanks very much for Jeremy. Finally, once again, and we'll see you again. Okay. Thanks everybody. Bye bye.